Nucleic acids are the group of organic molecules involved with organisms' genetics. They're large polymers of repeating nucleotides, which are the monomers. So recall that all organic molecules, except for lipids, are polymers made up of repeating subunits called monomers. And in this case, they're the nucleotides. We'll talk more about those in a moment. Now, what you see spinning around here to the right is a small portion of a molecule of DNA, deoxyribonucleic acid. And DNA is the molecule inside our nuclei that carry the genetic code. Now, there's another group of nucleic acids called the ribonucleic acids, RNA, and they're also involved in what we call gene expression, actually reading and making functional protein product from DNA. So let's take a look at the monomer of nucleic acids, the nucleotide, and there's three general parts of a nucleotide to be familiar with. The first is a phosphate group. So you see here a phosphorus atom surrounded by oxygens there in the purple circle. And then there's a 5-carbon sugar. And chemists often number their carbons 1, 2, 3, 4, and 5 here, making up that 5-carbon sugar. And then finally, there's always a nitrogen-containing base. And these three parts make up a nucleotide, a single nucleotide, and these repeat over and over and over again. Now, you'll often see nucleotides represented in a very simplistic way, like you see at the bottom here, with the phosphorus group, the 5-carbon sugar, and the nitrogen-containing base. Now, this G here indicates the identity of the base, which can change, and we'll take a look at that in just a second. Okay, so let's take a look at this overall structure of DNA. And I want to break down this figure for you into smaller pieces here. So first, notice the repeating nucleotides, the structure we just looked at, and how it repeats over and over and over again. So again, you see the phosphorus, the phosphate group, the 5-carbon sugar, and the nitrogen-containing base, over and over and over again. And now notice that the sugar of one nucleotide bonds with the phosphate group of another nucleotide over and over again to create what we call sugar-phosphate backbone of this molecule. And then you can see that the bases stick out from that sugar-phosphate backbone and extend out. And you can also see here's one base of one strand of DNA interacting with the other strand of DNA and the base sticking out from it. And what you see here is hydrogen bonding. Remember those electrostatic interactions between the hydrogen of one molecule and the oxygen, or in some cases the nitrogen of another. And now I want you to notice in these boxes here the identities of the nitrogen-containing bases are different in some cases. There are 4 total different nitrogen-containing bases: adenine, thymine, guanine, and cytosine. Okay, so again, take a look at this figure and then familiarize yourself with the structure once again. Notice that the sugar-phosphate backbones are similar to the railings of a ladder, with the hydrogen-bonded bases jutting out in between, kind of like the rungs of a ladder. And indeed, you'll often see this ladder-type formation in pictures of DNA. Now, in the actual molecule, those two sugar-phosphate backbones wrap around each other into the familiar double helix form of DNA, that sort of twisted ladder appearance. So again, you can see here the hydrogen-bonded bases jutting out in the middle and the sugar-phosphate backbones winding around each other. Okay, RNA, the other kind of nucleic acid, remember this is ribonucleic acid, is much the same as DNA with a few important differences. First, notice you have the phosphate group, 5-carbon sugar, and the nitrogen-containing base, making up the one single nucleotide monomer that repeats over and over and over again. However, some of the differences include the fact that RNA is only a single strand. There is no hydrogen bonding to another strand, as there is in DNA. Also, the difference indicated in the name of the molecule has to do with the kind of sugar there is. In RNA, it's ribose, and it has to do with this hydroxyl group right here. So in DNA, this oxygen and hydrogen are missing, and thus that is called deoxyribonucleic acid, or one less oxygen in its sugar. And finally, you'll notice that there's another kind of a base here called uracil, and that's there instead of thymine. So in RNA, you'll see U's instead of T's. You will never see any T's in the RNA sequence. And finally, the genetic code that our DNA carries refers to exactly what the sequence is of those nitrogen-containing bases, the C's, T's, A's, and G's. They can be in any kind of order whatsoever. And the order of those bases determines which order amino acids are placed in when our DNA is read. So amino acids are the monomers of protein, therefore our DNA codes for what order amino acids need to be put in to build the proteins and the enzymes of our body. These sequences, the genetic code, of course, is very different among different species and is even very different within among the individuals within a species. Thus, we all have a different DNA sequence.